Good morning, everyone. In the last class, we have had an introduction to screening. So in the previous classes, we have discussed about gene libraries. We have discussed about several protocols on cDNA libraries as well. So what we are going to discuss now is how to select the clone, clone of interest from the whole of it. So usually we will have at least two of them. Maybe most of it is by colony hybridization or plaque lifting that we will discuss in the coming slides. So let's get in there. The objective you should remember is first, we should try to know, say we have several clones and we want to figure out which clone is having gene X, for example, right? So how do we find it out? We can use hybridization using for nucleotide sequence based on nucleotide sequence. We can also use immunochemical, that is using antibodies if you are recognizing at the level of protein. Okay. So the first one is in general, or I can actually skip this, but I hope you realize that we will use this one. So here is a gene library. It is of several clones. Each clone must be having a gene construct. It could be having a gene A, gene B. This one could be having gene C, and so on. So what we will do is from each clone, individual clones, we can isolate the plasmid and then digest it with BAM-H1. That we are assuming if the vector used is a plasmid here, or assume it's a plasmid, and the gene library is made using this plasmid at BAM-H1 site. Okay. So, and once we have isolated plasmid, digested with BAM-H1, and then if we run each of these clones, like this clone 1, 2, 3, and so on, and then we would get a fragment of the vector, right? It will be common in all of these clones because all of them must be having this vector. And then they will have a fragment different in each of these cases, which is because of the different fragments present in different vectors, because this is a gene library. Okay. This is a good way of assessing the quality. Say, for example, I have a theoretically another one, something like this. And if I get the same vector band in all, that is good. To at least we know that it is the vector. A vector is present in all the clones. And if we find something like this, okay, what does it indicate? It indicates the likelihood that it is the same fragment in all of them. So when we are doing this, sometimes it may also occur that some fragments are overly represented in a gene library and some fragments are underrepresented. So this would be a bad quality. Or for example, if you find, the easiest way of understanding is if you made a library and you have isolated the plasmid and then you have digested it and then you find only vector fragment here like this, that means it's a bad quality. There are no fragments inserted into the vector. It's vector-vector religation. That is a way of how you can identify or assess the quality of the gene library that is being made. Okay, now we are coming to screening, right? We want to find out which one is the gene of interest, and how do we know? If we know the gene sequence, then you can design probes, or you can design PCR primers also, right? And then based on using either hybridization techniques like Southern blotting, you can identify the probes. We can do, if we know, we can use probes to identify Southern blotting. If we know the sequence, we can also use PCR primers that are specific to the region of our interest. And then we can probably figure that out. But that's not always possible. Sometimes, say for example, we have a cell, and we spoke about amylase, for example, a few classes ago. This is a novel amylase which has different sequence and so on. All we know is its enzymatic action, that it hydrolyzes glycogen, say, or starch maybe. And so we can take this amylase, find out the protein sequence using some of the techniques up there for sequencing the proteins as well. Again, by Sanger's method also. Sanger, I think, received twice Nobel Prize, one for protein sequencing and one for DNA sequencing. I think so. You should look it up. Anyway, we can find out a protein, a part, at least in some parts, we can find out the protein sequence. So if you know protein sequence, you can use two ways. One of it is if you have antibodies against the protein, then we can use immunochemical methods. If we don't have antibodies, then we have to go with functional studies. That is also possible. Like, say for example, if it is amylase, we would know that if amylase is produced, and assume it is secreted also in the gene library that we have constructed, then it would have been producing it will have the zone of clearance with the iodine test. That is another way of you can find it out. There are also complementation assays, or where if you have chosen host that is deficient in some aspect, say you did not provide any, the host is deficient in using amylose, and you provide only only amylose as a carbon source and energy source, just theoretically, then the cell that has got the amylase gene, the clone that has the amylase gene, will grow better than the others. So that is the functional or metabolic complementation. You can think it of that way. There will be a problem. Say, the objective is to identify the amylase gene. The problem is the host cannot, say it is a eukaryotic gene, so it might be coming with its own, with the introns, right? And if you're using a host like E. coli does not have splicing machinery, so there is no possibility of a protein production, right? Protein production is compromised. In that case, how do we identify? We will have to go with nucleotide sequence, like the hybridization techniques. It's good to go with hybridization technique, but we have a problem. We have a problem is that we know the protein sequence, we know the protein sequence, but we don't know the nucleotide sequence. For hybridization, to design a probe, we need the sequence to make. So here is one of the protocols somebody has used, and I like it. So all these people knew was the amino acid sequence like this. And based on that, one can make the nucleotide, a probe based on kind of reverse translation. I mean, it is just on the information level. So you can choose regions which are consisting of amino acid stretches which are coded by lesser number of codons. Say, for example, I think his is coded by CAT and CAC, phenylalanine TTT, TTC, and proline, I think, is coded by 4 codons, phenylalanine 2 codons, and ATG methionine. So they are going to make a probe. Okay. in this particular case, any one of the sequence is correct, right? Not all of them. So CAT, so you can have a what you call a mixture of probes. Like we discussed about yesterday, how a gene synthesis is basically made. And I also shared a video, YouTube video on the Google Classroom. Please check it out. And it's an interesting, or at least it will give you a fundamental concept about how gene synthesis is made. So here I would put it as simple. Again, I will repeat in a very simplistic way. We don't have to mug it up, but I think it will be useful if you can remember it. So I want to make a probe, and I want you to tell me how it should be done. First, I will add C. And what should I add next? So remember, my goal is to make a mixture of probes, which all of which would be actually coding, which would be coding for this peptide. So what mixture should I get? Next, second base is adenine, right? So I will add, yes, please, go ahead. What should be added?
Adenine, sir.
Yes, ATP with the locked 3' OH we should add. So A will get polymerized here or added on, and then we will wash. And what should be done for the next one? What should be added? Next base for the third one?
Add both
We should
D
add a
and C.
mixture of, yes, add a mixture of TTP, DTTP, and DCTP. So what will happen? In some cases, T might be added. In some cases, C might be added. Right? We have already, just by the base, we have two different nucleotides or oligonucleotides coming up. Next, we should add the fourth, fifth are T and T. So we will just add T. After that, I will wash and then T again. And now we have to add T and C. So we will add T and C, a mixture of them together in one addition step. So some of them will get T and some of them will get C. Again, the diversity would have increased, isn't it? Right? So now we have 4 different types of oligonucleotides that are coming up. And that way, we can slowly build the whole nucleotide diversity. Each round of cycle we have, we go ahead. We will keep on getting. Next ones are CC in all the cases. And now we have to add 4, right? You will have a large number of all the DNTPs are added, so that you will generate a large number of or diverse probes. At least one of them would fit to this nucleotide sequence that is present in the gene that we are looking at, say amylase, right? This is primers are synthesized in serial order. Probes are also synthesized in order. Here, in this special case, we want a mixture of probes because we don't know the nucleotide sequence. We want to try all of them, and at least one of them would fit. And that is what the approach is. This is during the constraints where you know the amino acid sequence, but you don't know the nucleotide sequence. In that case, how do we approach is the question here. And this is the way of doing it by probe synthesis. You generate a mixture of probes and then use them for blotting techniques. Okay. So blotting techniques, how do we do the blotting is very much similar to that of Southern blotting, except there are simple differences, of course. So we would have plated the library on nutrient agar plates or LB plates. So they would have grown into each one colony. One clone represents, maybe it has one gene there in it. Now you put a filter on top of it, something like this. You touch it, and you remove something like replica plating, you're kind of, it is blotting also, right? On the filter, you will have these spots, each of them representing the colony that is present on the master plate. You can think of this as the master plate. And then we add, there are many steps which we will see in the next slide, but this is the general overview, where we will hybridize and detect the label. That means we will cut open we will digest the cells, open them up, denature the DNA, and add the probe. And then we will develop the radiogram and figure out which of these colonies is giving positive signal. So in this case, we should have known the sequence, nucleotide sequence. Only then we would have been able to generate the probe, right? So using that probe to whichever clone it is binding, we now know that colony, we know the location where it is because we have retained the original or the master plate. Then we can pick that clone and go for further characterization. This is simple way of saying of the technique colony hybridization. You're doing hybridization techniques and you're using the whole colony, so you call it as colony hybridization. This has more details in the, this is the similar protocol. It has more details. Probably we'll try to understand them. So, okay. So what we have is, this is the plate on which we had a replica plate made. This is, you can assume this as the master plate. And then we transferred them onto a filter or the nitrocellulose disc. We take the disc out, and then we take the disc, and then we will lyse the cells using an NaOH. We then, because it's alkaline, we will neutralize it. We will add proteinase to digest the proteins that are present in the cell so that the DNA is much more exposed. And then we will wash so that majority of the things or loose things will go off. And then we will bake it at 80 degrees centigrade. So the DNA that is present will be denatured. At the same time, it will also get bound to the, or fixed onto the nitrocellulose filter. Then we are going to add the probe, which is usually radio-labeled probe, and then we will perform autoradiography. In the autoradiography, we would get a signal, and we might, if one of the clones has the genes that we are looking for, it will give the signal. We will then take that. Using that spot, we will check the reference colony on the master plate, and then we will take it as a positive colony and for further characterization. Okay, so this is called as colony hybridization. Okay, not always we make libraries on plasmids. This is a plasmid-based gene library. Sometimes we make libraries based on phages. Phages do not form colonies, right? Bacterial cells form colonies. What phages form are plaques. They are cleared zones where the bacteria, they would have kind of parasite-like, they would have gone onto the cells, replicated and killed the cells, and they would have made plaques. So how do we hybridize in that scenario? How do you screen for the clone of interest in that scenario? This is called as a plaque lift method. It's almost the same, except here we have the situation, we have plaques instead of colonies. So we would have made a plate which has the plaques, and then we would have grown the plaques, and then we would have put the nitrocellulose sheet, very similar to what we have done with the colony hybridization. On top of it, we put, and then we take it up, take the nitrocellulose plate, and then we will add We will do the similar process. We don't need to do too much because this is plaques. Plaques have protein casing, maybe majority of them, and DNA. There is not much, not too complex. So there we would put it in alkali conditions, neutralize, and then we will have 80 degrees centigrades for baking. That will allow the DNA denaturation and also binding to the nitrocellulose filter fixing kind of thing. Then we would have used a probe for hybridization. Again, we should have known the nucleotide sequence, and then that probe would be used, washed, and then autoradiography. We will then know in which plaques, which of these plaques that are present in the plate actually have the gene of or clone of interest. And when we will take those plaques, grow them in E. coli, and then save or go for further steps. So both are very similar techniques. One of it is colony hybridization, and this one is the plaque lift method. Okay. Yeah, we have already seen this, but what we have put here is how to say the goal sometimes is we want to put the gene library in order. That means, say, for example, A, C, D, E, F, Q. These are the genes, and these are the genes. This is the order of the genes on a chromosome. How can we figure out that A is beside C, and can we figure out C is beside D, and so on? It is something like chromosome mapping. What is restriction mapping? When we discussed about restriction mapping, we were trying to figure out the restriction sites that are present on a given DNA, and also the distances between the restriction sites, right? That is what we meant by restriction mapping. Here, chromosome mapping is usually or mostly about what gene is present, the order of the genes. When we constructed gene library, we wanted to, one of the at least the goals are at the system level, if you think of as the genome, we want to find the order of genes. We want to also know the nucleotide sequence of the genome as well. So how can we say we have made the gene library, and how can we find out the order? Then we can probably put it into into proper way, and we can probably have a fairly good idea about the map of the chromosome or the genome. There are two types of genomic libraries. One of it is overlapping, and the other one is non-overlapping. I hope you remember when we construct gene library, the genomic DNA is often partially digested. Yes, it's partially digested. It is not partially digested because we want very long DNA fragments. We partially digest so that, say, if each of these is a genome, each of these lines is indicating the genome. And if we make full digestions, we would have had all of them are cut in this way. And there is no way of knowing which fragment comes after which fragment or gene, say, for example, A, B, C, D, E. There is no way of figuring out how A is besides B. Instead, if we have made partial digestions, right, because of which, if you are making partial digestions, then we will have, then we will get overlapping fragments. Like this. And then there is a way to figure out which is beside, which gene is present beside which gene. Because when we make a partial digestion, say gene A, B, C, D, and E. In this case, A alone can be cloned in one fragment, right? Now look at the other one here, A and B. Those two will be cloned together in one clone. We can also find B and C are here. They are together, they are getting cloned into one of the vectors. Okay, so now let's fuse these, the colony hybridization and gene library, and see if we can figure out the order of the genes and put the gene library in a particular order. So I hope you understood this as well. There is a technique called as chromosome walking. Some of you might have already listened to it or heard about it. It is about making serial colony hybridization. It is a colony hybridization technique, and when I say serial, you will do one after another, like what is shown here. Say, for example, I know a small sequence of, I know sequence of gene A, but I don't know anything about the rest of the chromosome. How can I map the genes that are present? And how can I know the order of genes that are present? So because I know the sequence, I can synthesize a probe and call it probe 1. And then I will plate these colonies, and each of them has a fragment from the genome. I would use probe 1 to figure out and figure out whichever has it, assume that this is the one that has gene A. Okay? Gene A may be like this, and also may be having something else sometimes, right? Say, for example, the probe is binding here in this clone, in the fragment. Now, I can take that clone and figure out what is present on this side of the clone. I can figure out the nucleotide sequence. If I know the nucleotide sequence of this region, I can design a new probe, right? And then I can figure out or use the whole clone itself as the fragment itself as a probe, and figure out which other clone is sharing some parts of the gene of the genome. And here we will find the next clone. And now you can take clone 2, use it as a probe, the fragment, and then figure out which all of these colonies share these sequences similar to that of clone 2, right? So that means we can say slowly what is happening is that we can slowly build the order of genes. So the first, we will, I mean, when we are making an order, of course, we will give it as whichever we start with, we'll call it as clone 1. And that has similarities with clone 2. That may have similarity with clone 3, and so on. Then we are putting the gene library into an order. This technique is called as chromosome walking. You are walking, it is something like you're taking one step, finding out something, and then taking another step and another step. This is called as chromosome walking. Yeah, there are some other names you might have heard, and this is chromosome jumping is there. Don't confuse it with jumping genes. Chromosome jumping is there. I hope you also heard about footprint, which we will discuss in this course again. These are some of the names. It indicates it's better if you can associate the principle with the name always. Okay, did you understand this?
Yes, sir.
Yes. Okay, good. If you're writing CSAR and or GATE, some of the questions that will come is mapping using conjugation. interrupted. This is a basic science thing. It's not as an application, but mapping using conjugation, like in the interrupted mating experiments. You can look at Pearce or any genetics book, and some of the classical experiments, bacterial and viral genetic systems. I think this is the title of the chapter as well. These are some of the important questions that will come in CSAR, because they are classical experiments, and majority of the classical experiments have not necessarily the great thing about technology, but it's about logic. Technology we have to memorize, and that's about it, measured most of the times. And instead, in the logic based ones, that is where your skill and your aptitude comes into play. And that's why I think they do ask a lot of questions based on classical experiments. So please have a look at it, and if you have any questions, we can discuss in some class. Okay. Yes. So this is how we make an ordered library, right? Colony 1 hybridizes to colony 2, 2 hybridizes with colony 3, and 3 hybridizes with colony 4. Now we are going to go ahead with something called as subtractive library, and we will discuss race in the next class maybe. Let's see. Subtractive library is, I have two different cells, cell A and cell B. It is usually about cDNA library, but there are also subtractive gene libraries also there. But we'll just go with cDNA library. It's simpler, easier, and popular actually compared to the subtractive gene library. So both of these cells, assume, are from the same species. Only one thing, the difference is their condition is somewhat different. That is, say for example, we are growing, this is the control, and B is we are treating with some compound, we are treating with compound X. Okay, we have added some X compound in the growth condition, and in the control we did not add. Okay, so this is the test. I want to know the effect of compound X. I want to know the effect of compound X based on this. What kind of transcriptomic changes is it bringing in B? So we will have genes that are common in both the cells, which are usually the housekeeping genes or the cell lineage specific genes. There might be some genes that are expressed here but not expressed here because of maybe the compound X. Compound X might be causing some reduction in the expression of some cells, we don't know. Or you might not have some expression of some genes in A while there is expression in genes in B. If we say that these two cells are actually the same cells from same organism also, then their genome is same genome. What is different in this experiment is one of it is treated with compound X. So these two, the differences that you will see in the transcriptome and also in the proteome, but we are not bothered about proteome at this stage. The differences in the transcriptome must have been because of compound X, isn't it? Otherwise, we are saying same cell type from the same organism with the same genome should have the same transcriptome if it is grown in the same culture medium. But this is one difference is compound X. So now I want to clone the genes that are expressed in B but not expressed in A. That is subtractive library. I want to clone the genes expressed in B, but not in A. So you will have all the B genes being expression. You are subtracting A expressed genes. What will be left? Genes expressed in B, but not in A. And that's why we call it as subtractive library. And because we are cloning them, we call it as a library. So here is the setup. You can consider this to be the A, that like in our experimental setup that I discussed. I've given only an experimental setup. It could be a same organism from or two individuals of the same organism. One is wild type and one is a mutant. It could be that way. It could be like the treatment and untreated. And treated is one way. You could also have tested in different developmental stages or cell types, um, cell type P and cell type Q. You could also have had it in cell that is in at the stage of morula and at the stage of some other cell at a later developmental stage. So here we'll go with the experimental setup that we discussed here. We have two cells, A and B we are calling them. Both are same species, same organism, same cell type. Only difference is one is treated with compound X. That is how usually drug testing can also be done. So driver would be A, that is the kind of control. And tester is B, that is the treated one in our experimental setup. Right? We call it the tester. What are we doing? We are subtracting A out of B. Okay, so what will be remain is B specific genes, meaning that they would not have been expressed in A. So we take the two cells, isolate the mRNA from both of them. The control ones, we would call them as driver mRNA, and the other one is tester mRNA, the one that is treated here, from which we are subtracting. This would be the tester, and this would be the driver. So we would prepare cDNA for both of them. And while preparing cDNA, we would have done something here. For the driver, we would have added a compound like a biotin so that it will be easier for us to purify at some stage. So we have cDNA from both the cells. The driver mRNA is conjugated with the biotin, whereas tester mRNA has nothing like that. Only cDNA is present, double-stranded. Now we are mixing both these cDNA. You mix them, you denature and reanneal. So let's see, assume these are the genes, cDNA from the tester, and these are the cDNA from the driver. Okay, this is driver and this is tester. And I will also give some gene names so that it will be easier. X, Y, Z, X, M, R. Okay, XMR I have given. Now we have mixed them and we have denatured and allowed reannealing. What would happen in that case? You can have, what are the possibilities? We should see the possibilities, right? X can find its another X and base pair like this. That is a possibility. There is also a possibility that the X from the driver can find its complementary pair and bind like this. There is also a possibility that X, one strand from the tester and one strand from the driver can bind like this, right? These are the combinations that will happen. We'll also check what will happen for M, and for M we will see. This is M, and there is no complementary sequence from X, right? So when we denature and let it reanneal, it will form by itself again like that, right? M will remain as such. So if we recollect, we have added a biotin here, and I will put the biotin like this now. So this has biotin, and this one too. And the hybrid ones will also have biotin like this. There are two things that do not get biotin. Which are those? This one and this one. Let's look at the next step. These are the three different types of combinations: driver, driver, driver and tester, like what we have gotten here. And we'd also get unique testers, some of them which do not get anything at all. But this is also a possibility. We should somehow be able to eliminate the formation of those. Although it is common in both, there is a possibility of formation like this. So what usually they do is they take 10 times more of the driver cDNA. When you take 10 times more, what will happen then? The probability of these forming will become very, very less, or we are decreasing the probability of tester mRNA binding like this. So all of them would form, majority of them would form something like this, because this is a possibility, and this is also a possibility because we have 10x times of the tester mRNA. So this is a very rarity. But in the case of M, there is no complementary in the tester. So it will remain as such. Okay. And now when we pass it through a column containing streptavidin, which has high affinity for biotin, all the drivers and the driver tester, the hybrid ones, they will bind, and the unique clones, such as in this case, if we pass, this will get the XX will get bound with the streptavidin. This will also get bound with the streptavidin. Sorry, we wouldn't have this here, right? Because we didn't add, yes. And the only one that will flow through will be MM, right? We will get this one like that. And then now we can go ahead with cloning procedures and then make a library. What is that library? That is a library of subtractive cDNA library. Okay. Do you have any questions on this? Please let me know. I hope you understood the objective of it. Objective is subtraction and cloning. I want B-specific genes. That means genes that are expressed in B but not in A. I want to clone them. That is what is subtractive cDNA library. We do that by mixing, denaturing, reannealing. We use the principle of hybridization and eliminating all those that are common with the driver mRNA. And how do we eliminate them? We eliminate them using some form of affinity. Here, the example is about biotin and streptavidin. So anything that is common or shares homology with the cDNA of the driver are subtracted. The remaining ones are the unique tester cDNA, and those are the ones that we will clone. Okay, that is called as subtractive cDNA library. Okay, let me know if you have any questions.
Sir?
Yes, please, go ahead. Yes, please.
Sir, how the driver and tester can base pair, sir? Because they are different genes, no, sir?
No, these are mRNAs, right? They are, okay, if I said there is a gene X in the tester and gene Y in the driver, well, gene X in the driver also. Let me go ahead with this. So I will just put it, I'll go a little more elaborate. I hope it's all right. So I will go with X, Y, Z. And in the other one, we will have X, Y, Z. Okay. Both are, the genomes are same in both the cases. What is different in the experimental setup that I have discussed, that is the only thing I'm talking about. So this is, we will consider as A, and this one we will consider as B. So in A, maybe X is expressed. Okay, I'll just put it as XX. Sorry, I'll use blue. This is X, and this is X. It is a mRNA, right? We'll just put it as mRNA. X is expressed, Y is also expressed. Z is not expressed. We'll just think of new Y. In the case of B, X is expressed, Y is not expressed, and Z is expressed. Okay. Now what are we doing? We are going to make cDNA of both of them. We will make cDNA of, the sequence is same, isn't it? X, gene X and gene X, mRNA X and mRNA X. The sequence is same. And then we get cDNA. Yes, this is X and this is X. The nucleotide sequence is same in between these two. Okay, so if I have mixed these two, and they would have denatured like this. And if I had this, the sample from A 10 times more in concentration, and if I allow, say, for example, this strand, the top strand, is complementary to the bottom strand here, and is also complementary to the bottom strand here, isn't it? So it can base pair with this, with the bottom strand here, this one, or it can base pair with this one. So what will happen, what can happen if we have taken excess, then they would base pair like this. The nucleotide sequence is same. Okay. And if you, yes, Ramya, go ahead.
Yes, sir. Clear, sir.
Okay. I'll just finish off with this one anyway, if there is any question. In the case of Z, we have this, there is no counterpart here. So when we mix denature and reanneal, it will form ZZ back like this. So they will not, and then using streptavidin, we will get rid of these, and what we will be left with is the Z, and that is what we will be able to clone. Okay, so the objective of subtractive cDNA library is to clone the genes that are expressed in the test compared to or relative to the wild type or the control. We don't want to clone the whole library because it will become a very big one. We just want to clone only the difference between two samples. Okay, there is, okay, I'll stop here. Maybe I think we're running short of time. Any questions? In the next class, I will take differential screening and immunological screening and race. Yes. Any questions?
Sir.
Yes, please.
Sir, I have a doubt in chromosome walking, sir.
Yes, please. Tell
Sir, in this, with the help of probe 1, how can we find the remaining sequence of the clone 1, sir?
Okay. Here, usually when we figured out clone 1, we will take out and we can go for sequencing. Okay. And if we know the sequence, if we have determined the sequence of this part, then we would have been able to make probe 2, a new probe. Or simply, I will just take the fragment from clone 1. Clone 1 would have been something like this. I would have taken this, radio labeled it, and used the whole fragment as a probe. As a probe, and that is what I think they have used clone 1 as a probe and figured out what is in following clone. And then they would have used this whole clone as a probe, I mean, the fragment, and then figured out the next one. That way, they are walking from one step to the other step. For the next class, I will try to design a problem on this. I usually do that. Today, I didn't make one. And we will practice it in the classroom so that it will allow the understanding much better. Okay?
So thank you, sir.
Yes, you're welcome. Anybody else? Any questions?
So I just have a follow-up doubt on the previous statement that you made.
Yes. Yes.
So you mentioned that the part of the sequence of the clone one can directly be used as the probe.
Yes.
But shouldn't we need the complementary one so that it can go bind with the clone too?
Yeah. You meant to say, for example, what you said is complementary is not a problem as long as the sample has both the strands, isn't it? If it was mRNA, we are using gene library. We are doing it at, even if you take the top strand or the bottom strand, you will have the complementary region within the genome. Both will get recognized. So either you recognize this part or you recognize this part, we know that the gene is present there. Yes?
Okay, sir. So do we
If
send
it is,
the denatured
yes, tell me.
DNA there?
Yes, say it again. I can't hear you.
Do we add the denatured DNA of clone one over there? Like send it as two different strands and then see where it is going and binding?
Usually, I think I removed one of the slides because I thought it is too much. During the hybridization, okay, we will maintain, there is something called as stringent conditions we will use, in which we will also have a temperature as a factor. Okay. And there is also washing step with the stringency with which we wash. So we don't necessarily, they are not in normal room temperature. They 45 or 50 degrees also can be maintained. And in that case, we can expect that they will be separate. And in the case of probe, the question is if we are giving a double-stranded DNA or single-stranded DNA. Is that right?
Yes, sir.
Yes. Yeah, I think we can see developing the probe is not a big deal actually, because we have discussed about in vitro transcription and in vitro translation, right? We could have used that clone to, and if we had a probe, if we had this clone and say T7 polymerase, we can develop RNA probe very easily from it, single-stranded, no issues at all. Okay, we don't need to know the sequence also. All we need is somehow be able to transcribe it, in vitro transcription be performed, so we will get radio-labeled RNA probe. I think this is how usually when I
Yes,
did
sir.
not chromosome walking but northern blotting, this is how we make the probe. But that's a good point.
Thanks,
That
sir.
if it is double-stranded, it is a problem. So I think this is a more or a better one. Okay.
Yes, sir. It's clear now. Thank you.
Okay. Yes. Thank you. Anybody else? Any questions? Okay. Thank you for attending. Take care and have fun. Please watch the video that I shared on Google Classroom. Yeah. Take care. Bye.
Sure, sir. Also, there is
Yes.
not